Good evening, everyone. Thank you for joining our 2024 virtual public meeting on chronic wasting disease. We'll get started in a couple of minutes, but first we're going to release a quick poll to get a sense of who's in the audience tonight. And I'm going to orient you to some of the Zoom features. So if you have questions throughout the program, please place them in the Q&A block, which should appear at the bottom of your screen. Some of our behind-the-scenes staff will be answering these throughout the program, and perhaps saving some for further discussion at the end of my prepared presentation. If you see your questions already in the block, you can upvote them by giving them a thumbs up rather than entering it again. And finally, if you find yourself having technical difficulties seeing or hearing this presentation, you can raise your hand and our staff will try to assist you. Keep in mind that this presentation is also being recorded, so if you have to jump off early or you want to revisit any of the content tonight, you'll be able to do so through our website and our YouTube channel, and it'll be posted in about a week from today and be publicly viewable. Okay, with those items out of the way, we will begin. So thank you again all so much for joining me tonight. My name is Rachel Rudin. I'm the state wildlife veterinarian for the Iowa Department of Natural Resources. I like to say that about 40% of my time is dedicated to this disease, chronic wasting disease, which we'll be talking about at length tonight. And you'll see why. It's because of the importance and the complexity of this disease. So we'll definitely capture that flavor, I think, with the rest of the presentation. Okay, so already skipping ahead. Okay, so what is chronic wasting disease? So chronic wasting disease, or CWD, is a slowly progressive, uniformly fatal brain disease of cervids affecting white-tailed deer here in Iowa, but also wild mule deer, elk, reindeer, and moose. It belongs to a group of diseases known as the transmissible spongiform encephalopathies, which may include some familiar diseases like scrapie in sheep, and bovine spongiform encephalopathy, or mad cow disease in cattle, as well as Creutzfeldt-Jakob disease in humans. And this group of diseases is spread by a misfolded host protein called a prion. Now, healthy prion proteins should be expressed throughout the body of vertebrate animals, but when they take on this misfolded or alternate shape, they also acquire an infectious property that allows them to self-propagate and cause disease. So it's important to distinguish them from some of the more common disease-causing agents like viruses, fungi, and bacteria. So I thought we'd start with just kind of the big picture in North America. Iowa is not unique in their plight with chronic wasting disease. We're one of 35 states and 4 Canadian provinces that have CWD circulating in its wild cervid populations. There are additional diseased animals or disease populations in Scandinavian countries, including Norway, Finland, and Sweden, as well as captive elk in South Korea. So how is this disease spread? So primarily we're worried about direct transmission, or deer to deer, through the contact with bodily fluids like mucus, saliva, and urine as part of their normal day-to-day interactions within their communities. There's another component that definitely plays a role, which involves environmental transmission or indirect transmission, and that's through the contamination of areas. This may include, feeding stations, soil or scrape sites, licking branches, things like that, that get contaminated with fluids from infected animals and then can spread disease when the next animal comes into contact with them. Just a bit about disease progression, and really one of the most challenging parts of this disease. So once an animal's infected, it can take 1.5 years to up to 3 years for them to begin to show clinical signs of disease. Now, during that time, most of that time, they're actually infectious to other animals. So they start shedding saliva as early as 3 months following infection, and in their urine as soon as 6 months. Another important aspect of chronic wasting disease is that the prions do accumulate in all tissues, including the muscle or meat, antler velvet, and the skin. Now, prions are highly resistant to deactivation And we know that they can persist in other disease systems for years, if not decades, and remain infectious. An overview of the clinical signs. So in a wild population, we oftentimes just have a snapshot. Now, sometimes with our trail cameras, we get many snapshots, or if they're attending, your bird feeder, you can kind of track an individual over time. But most of the time, we're not going to see increases in urination or salivation, unless they're doing it at the time that they're being observed. But things like progressive weight loss get reported quite frequently. So I hear reports of skinny deer a lot, but there's lots of causes for skinny deer. And again, we wouldn't expect a deer in that long 1.5 to 3 year asymptomatic phase to show any clinical signs at all. And so with all that, we cannot diagnose CWD on the hoof. We really need to test the animal. And we do so by pulling a lymph node that's behind the lower jaw. Keeping in mind that in Iowa, most of the deer that have tested positive to date have been, otherwise they looked healthy. They were in normal mentation, behaving normally, and they had good body condition, and oftentimes large racks if they were adult bucks. So we get questions about that quite a bit too, is would they still be able to build a rack? And it turns out that's something that kind of stays. That's one of those survival mechanisms in a deer society. And so because we can't tell that a deer is diseased by looking at it or looking at the tissue, we need to test it. To make informed consumption decisions, we really do need to collect the lymph node and test it for this disease. And we follow the Centers for Disease Control and Prevention's recommendations that deer harvested in areas where we know CWD to occur should be tested. And then the meat from positive animals should be discarded. So that means that if you're intending to share that meat with friends and family, you might want to hold off until you have those test results in hand. So here, we'll go through a couple of slides of actually the same base map of just color coding. The light gray means that we haven't detected CWD in those counties. The dark gray is places where we have detected the disease. And since we last sort of talked at our 2023 virtual public meeting, unfortunately, a lot has changed in the state of Iowa. So we've had first detections in 8 new counties during last year's surveillance period. So 23 to 24 surveillance season, we picked up 8 counties. They're listed here with the little lightning bolts. And you can see they really have no rhyme or reason. So we have several detections in central Iowa, we're expanding the disease front in northeast Iowa, and some really interesting detections down in southeast Iowa for the first time. Now, so far for the 24 to 25 surveillance season, we have additionally found 3 new counties, those being Shelby, Wapello, and Davis. So we're seeing that CWD is doing what CWD does, and it's spreading geographically and increasing in prevalence locally. But something that's really important and I want to emphasize is that most of these counties where we've had CWD occur, so we have 26 counties where we've had detections. Most of them, we've only had a single detection. So that means that the disease is definitely moving around our landscape. We know that deer move around our landscape, so that all makes sense. But it doesn't seem like it has taken root in all of those places. And that's a good thing. It means that our interventions can really have an effect if done early. And so I wanted to give an overview of what happens once we have a first detection in a new county. We increase our sampling and surveillance in that county for the next 3 to 5 years. And so we're looking to achieve 750 total sampling points. And this is what we use. We use a point system in places where either the disease isn't known to occur or we know it has occurred, but only with 1 detection. And you'll see in the next couple of slides what the point system looks like for different age and sex classes, but bucks have the highest points. They are worth 3 points because they are the highest risk age class. And then we have yearlings does as the lowest point contributor at a 0.5 point. So we'll go into that a little bit more in the next slide. But if we do have additional detections, that will transition this surveillance area into a disease management zone, where we will then be looking at estimating prevalence or the proportion of diseased animals in that population locally. We may adjust harvest goals, increase harvest opportunities. But if we don't find additional detections, we actually will just return back to routine surveillance. So we know, especially with all of these counties coming online with single detections, that hopefully they're not going to result in new endemic areas or places where we expect the disease to occur. And so they will go back to our surveillance system prior to that first detection. And of course, our goal throughout the state is to maintain a population that's at that low end of our population goal, but still offering a quality recreational experience. So balancing the many needs and interests of deer hunters, and the resource. So here is our current sampling quota map. And you can see in the shades of tan and brown are the county surveillance quotas. So we do collect samples throughout the state from all counties. And those are represented in those tan and brown. The green areas indicate places with targeted surveillance. So either they're the rest of the county that contains a deer management zone, or they're in places where we've had that first or initial detection, and we're trying to figure out more about what's going on in that area. And then finally, the yellow indicates endemic areas where we have established CWD deer management zones, and they're kind of these core areas where we know diseased deer persist. And at the bottom of this map, you can see the point quotas and the allocation. So the point values, adult bucks are 3, adult does are worth 2 points, yearling bucks 1, and yearling does 0.5 point. And that's based on who is most likely to interact with multiple deer and then has the highest risk of acquiring infection. And so adults who are servicing lots of females, sparring with bucks, they contribute the most value to our surveillance system. You'll note that fawns are not listed here because they're just on the landscape for such a short time by the time we're sampling. And so what happened this year with our new detections in Davis County, we increased the quota in that county to 150 points. The Wapello detection actually already fell within a surveillance zone, and so we didn't change that this year. And then the Shelby County detection, we established a new surveillance zone there with 100 points to see, what's going on, to get a better feel for of the disease dynamics in that area. And so the next couple of slides, we'll go over some things that you can do as a informed citizen in the state of Iowa or hunting in Iowa. And so, I mentioned our risk landscape for CWD, it really is changing in Iowa. And you can track what those changes look like by going on and checking our CWD interactive dashboard. I listed the short link here. It's just /CWD results. And there's a wealth of information here. So there's historic data, there's the current sampling season. You can look at trends in different demographic classes, so age and sex, affected counties that might be of interest that you're hunting in or that you live in, and across the years, those trends. And so here's just a snapshot of what our interactive dashboard looks like. This is the second tab. So this is showing our current surveillance season, the 24 to 25 surveillance season. And you can see that so far we have 19 detections listed. You can see where they're mapped to, what counties they come from, and modifications as new detections come online, they'll be posted here. And this gets updated frequently, especially once we get into those shotgun seasons. Okay, another way that you can stay informed is to become a CWD ambassador. So this is a fairly new program we launched in 2020. And it's a collaborative educational opportunity between the Iowa DNR and the Iowa State University Extension Outreach. And it's a 3-week course that's pretty intensive. So I'm giving you, an overview on CWD. We'll get real in the weeds in this course. And that's why it requires 3 in-person sessions and 2 online sessions. But for people who want the details, this is definitely a place to go and seek out. And if you are interested, please feel free to reach out to me directly, and we can try to figure out where you are, if we've had a course there yet, if there's opportunity to have a course there in the future, and go from there. Okay, another strategy that we can take is limiting carcass movement. And so unlike some other diseases, one of them being hemorrhagic disease in deer, once a deer dies with chronic wasting disease, the carcass remains infectious to other deer. And so responsibly disposing of carcass parts, even if you don't know the deer to be positive, but especially if you do, is a great way to limit the spread of this disease, especially out of those affected counties. So responsible disposal might include landfill. That would be kind of the gold standard, but not all landfills accept your carcasses. And so burial might be an option if you plan ahead and have dug out a pit before the ground is frozen like it is now. And then, maybe the best option for you, depending on your area, will be to just leave it lie at the site of harvest. And so that's always an option. This map just depicts hunters that were successfully harvested a deer in 3 of our endemic zones, so Al-Maqui, Clayton, and Wayne, and then shows their home zip code. So you can see all those radiating lines coming out, and that means that those hunters hopefully were responsibly disposing of their deer carcasses. But if they were not, we have lots of exposure there with deer moving around our landscape, but by the means of people, not through their natural movement events. Another great option is to contribute samples. So that might seem obvious. Reporting sick deer or roadkill. So even if you're not someone who is going out to hunt, you can still pass a roadkill and tell us about it. So that is a great opportunity. And many of our first county detections have actually come from roadkill. So it's an important source for sure in our surveillance program. And if you see sick deer, so of course, those are another opportunity. And then if you are a hunter, you can test your harvest. And so you can check our interactive dashboard that I just showed you and check if we have open quotas. You can use Dropbox locations that are available around certain parts of the state, or arrange a time with the local management unit staff to have your deer sampled and contribute to our statewide surveillance quotas. You might also consider using the hunter submission pathway for a quicker turnaround, or if it just better suits your needs. And we'll go over both of those options a little bit. So nested on the left-hand side of this slide is how to find our CWD Deer Management Zone interactive dashboard. So it's a different dashboard. It's a little bit buried, but if you follow those steps of deer hunting, deer health, chronic wasting disease, down to CWD Deer Management Zones, you can find this interactive dashboard, and it'll show you the locations for sample drop-off freezers. So those are these little red icons here. So that's helpful, that you don't even need to, meet with someone. You can do it on your own time, your own schedule, and drop off a whole head to those locations. Now, just a bit about the hunter submission pathway. So this is a partnership with Iowa State's Veterinary Diagnostic Lab. Also, as maybe a disclosure or for reference, I oversee all of the testing at the Vet Diagnostic Lab. So I have an affiliate appointment there, and I monitor, all of our results coming out. So whether you're doing it through the hunter submission pathway or through our surveillance program, I'm the point of contact basically for these. So if you have issues, definitely reach out to me. But this is a partnership we started. It's really been blossoming in the past couple of years. And it allows you to test deer that either don't meet our surveillance criteria. So if you've harvested a fawn, but you want to get it tested, I already told you that it doesn't contribute much value to our statewide surveillance program because we're trying to leverage our resources and get the best information. But it can still tell you a lot about making an informed consumption decision, getting that fawn tested. So that's definitely a viable strategy to use, the hunter submission pathway. And then, of course, if you are hunting in a county or a DMC that has exceeded its quota, then you can always submit to the hunter submission pathway. And then other, options if you're harvesting out of state. So of course, we're talking about CWD in Iowa, but we know people go to surrounding states, maybe out to the west, harvesting things that aren't just white-tailed deer. So this is kind of a nice option for all those, that different variety that you might have. There is a cost. It's $25 per head. That is what we as the DNR pay to get our samples tested. So that same cost would then go onto the hunter. But there is a quick turnaround. So usually we're seeing results turned around in 1 to 2 days, which at times, especially, following a shotgun season, or, opening weekend, like we have coming up, there could be some delays. We try to hit 2 weeks turnaround from the time the lab has received the sample to the time it releases those results. But the hunter submission pathway might fit your needs better if time is the issue. Another strategy, ditch the feed and mineral. So encouraging a natural congregation, crowding, and stress, basically using feed and mineral, it creates problems that wouldn't naturally occur in deer. We know that they're moving, we know that they're interacting, but it really can exacerbate an issue, especially in some of these endemic areas. And that added stress can make them more predisposed to acquiring infections, other diseases, including CWD. And then, of course, keep hunting. So this is a really important part of our traditions and culture here in Iowa. And it's also, honestly, our best deer management tool. So hunters help us manage deer populations. We all know that. And so keeping hunting keeps CWD, hopefully at those lowest thresholds. Okay, so with all that, we all have a part to play in this issue. It doesn't just touch hunters and landowners, it touches all different types of industries and individuals, wildlife watchers, wildlife enjoyers. So CWD is everyone's issue. And hopefully I've convinced you, I've made a case that we should do as much as we can in our own individual lives to limit the spread and definitely not encourage further spread. Okay, so that felt brief. It looks like we're under a half hour. So that might have been the quickest virtual public meeting we've had, but I really appreciate you attending. We'll move over to the question portion. If you haven't already, enter those into the Q&A block. If you have more nuanced questions in the weeds, please email me. I'm happy to field those questions. And like I said, I'm the wildlife veterinarian for the DNR. So my scope of responsibility extends beyond deer. So if you have issues moving forward, concerns about wildlife health, please reach out to me on those fronts as well. And with that, we'll transition to the questions. I'm going to try to access them. So just give me one moment. Okay. So, okay, we'll just go through the open Q&A questions, I think.
Rachel, there's been a lot of questions about people and the safety concerns with eating meat or not. If you could just talk about that a little bit, it'd be great.
Yeah. So that's the ultimate question of the concern with CWD is, can people eat those deer that test positive, and are there risks to their health? And that's an open question. So what I can say is that people are treating this very seriously. The CDC has a team that's actually looking at trend data. So it's not, there are a lot of collaboration and a lot of states, contributing data and that kind of thing. But ultimately, the question's still out. What we know about prion diseases, which make them so complex, is that what goes in when you have an animal that contracts CWD can be different than what goes out. And so we know from lots of experimental studies that can even mean expanding the host range, meaning what are the animals that are susceptible to a disease, that by cycling through more hosts, it can actually change that disease's ability to infect new hosts. So that's why there's so much concern around it. And why, if you know that your deer has tested positive, we would advocate for disposing of that meat. But ultimately, making an informed consumption decision means that if you still want to eat the animal, that's absolutely fine. We just want you to know kind of what those risks and how we can best characterize those risks are. So hopefully that clarifies some things. At this time, there's no known reports of people contracting CWD, but there have been some interesting studies with other hosts. So a lot of the experimental studies, they will actually inject, different species via brain inoculation. So really a natural route. One where we've seen that it's a much more natural route of exposure by just feeding venison was swine. So pigs consuming infected venison, they did actually contract the disease. And so, we know that it can extend beyond deer, but how much beyond is the open question. Okay. Chris, do you want to feed me another one?
Yeah, is there any known predators that have gotten CWD from deer?
Okay. So there's been some studies looking at some felids. So mountain lions, there were a pair that were fed greater than 50% infected venison in their diet per year for about 17 years. And they found no evidence of them contracting the disease, no signatures within any of their tissues, including the brain. Kind of complementary study maybe in bobcats showed that bobcats actually reduce the amount of prion in their, so through digestion, what goes into them, about 97% of the prions will actually be deactivated or undetectable otherwise when it comes out in their scat or feces. So we don't know of any carnivores or any predators contracting the disease. There were some movement studies looking at coyotes and crows that found that they can act as transport hosts, which would mean that there can be some viable prion or infectious prion that can move through their gut tract, that they could maybe introduce it to new areas. But neither of those species were really exposed to infectious prion over time to really answer that question of if they could be susceptible.
Okay, we've got a couple questions on test results and how fast we can expect those and what would affect those turnaround times.
Yeah. So like I said, we strive for a 2-week turnaround. We definitely saw some huge delays last year, and we've kind of worked out some of the pipeline a little bit better. So we are not anticipating that this year. So far, we've been hitting about 6 days from receipt to results. That was for the month of November. So it's something that we're tracking closely, and we have some options if we're starting to see things go off the rails. But we intend to get those results back to you within 2 weeks.
Okay, how about any safety concerns with processing deer and also like with cleaning cleaning deer? And also, can you talk a little bit about lockers and what some of them are requiring and some of them aren't, and just the things going on there?
Yeah. So in terms of cleaning, at least cleaning equipment, if that's what the question kind of is referring to. Um, so again, kind of leaning on the weight of evidence from experimental studies. So we know that a 40% bleach solution with a contact time of 5 minutes can deactivate prions on metal surfaces. So it was tested with a metal rod that was supposed to approximate a knife. So that's a pretty caustic level of bleach, but it's a short time. So you wouldn't want to leave your equipment in that level of exposure for, days or even an hour maybe, but 5 minutes should be okay. And then you know that you've really deactivated what it may have come in contact with. One thing to keep a note with any kind of disinfectant or disinfection protocol is that you really need to remove the organic material. So if there's a chunk of tissue, it's not going to necessarily deactivate anything in that chunk of tissue. So have warm soapy water, clean it ahead of time, and then put it in that 5% or the 5 minutes for 40% bleach solution. In terms of lockers and what they may require, so it seems like most lockers will accept tested deer, with the caveat being a lot of them want to know the results ahead of time. So what we advocate for in chronic wasting disease ambassadors, I think it's a good practice in this kind of world of uncertainty we're living in with CWD, is you can kind of do a rough processing of your deer, quarter it out, wait for your results, and then put in the rest of the effort to really further butcher it or bring it to a locker at that point. If you have the space and capacity and that kind of works out for you. There are lockers that will take tested deer for sure. You just need to kind of know ahead of time and kind of have that game plan ahead of time so you can figure out how you're going to store it in the interim if they don't accept tested deer. And one other kind of thing to think about with this whole question of lockers is commingling deer. So we know that lots of times we're not getting the deer that we turn in to a locker, or it might be, part of your deer and a part of 10 other people's. So considering if you're getting your deer tested, but you're going into a batch of deer that haven't been tested, you might not be, having that same confidence in what you're getting back as with what you went in. So just kind of running through all those outcomes.
Rachel, what are the options for people to take a sample? Either both, how could they get one done, or what resources are available so they could learn how to do it themselves?
Yeah, so that hunter submission pathway I mentioned, most of our resources or all of our resources really are there to learn how to take your own sample. So that's, you can go check that out at iowadnr.gov/hunter submission, the full words spelled out. And we have a sampling video there that we created in partnership with ISU Extension Outreach. We have a new sampling card, so kind of instructional card that also shows how you can age your deer, because we do factor in age to our surveillance data. And, it's something that we want to keep track of, and better educating people to use the teeth to age your deer will help in that effort. So that's a nice instructional card that kind of goes over what that video shows. Then you can turn to the video and see it in real time. CWD ambassadors, like I mentioned, one of the days, one of those in-person sessions is actually learning how to sample your deer. So if that interests you, but you want kind of a group learning experience, that might be an option. And then of course, staff across the state are sampling deer to reach the surveillance quotas for this year. And so if you have a harvest and you don't necessarily want to learn how to collect your own sample, and you definitely want to go through our surveillance system, then I would reach out to your local field staff. So that would be the wildlife biologist for your unit, the unit where your county is covered, and reach out to them about getting your sample tested.
All right, Rachel, could you talk a little bit? There was a question specifically about Colorado and how bad it is there. Could you just talk a little bit about other states and where the disease is worse or that kind of thing and what they're seeing?
Yeah. So I would say Colorado and like many of the Western populations, uh, it's more of a population structure difference. So the herds of, whether they're um, mule deer or elk or even, whitetails, they're just interacting differently. And in the Western states, they can be much more isolated and segregated to an extent. So there's less mixing across groups. And so there can be very high prevalences. There was a study that came out of Wyoming that found a 10% annual decline in abundance each year related directly to chronic wasting disease. But that was an affected herd. So there could be a herd in a different basin that's unaffected and has a low prevalence of disease, of CWD specifically. To put kind of where we are in perspective, so we had our first detection in 2013, but we started sampling in 2002 when two of our neighboring states, Illinois and Wisconsin, found their first positives. So we're about a decade behind in some regards. Those states, they've had very different management strategies, which is, maybe contributed to the apparent changes in prevalence there. But in the original county in Wisconsin, Iowa County, Wisconsin, an adult buck has a 50% chance of testing positive. Basically, so you might have, it's a turn of a quarter, whether you have a positive harvest or not. And the numbers of positives that they get in that state are hundreds or are actually thousands, can be over 1,000 each year. Whereas we're, we have maybe over 100, but we're still staying pretty low comparatively, if that helps. Okay, I'm just looking through.
I did see a question about, I think University of Minnesota is testing muscle and not just lymph nodes. Can you talk about the effectiveness of that or where that research is at?
Yeah. Okay. So I mentioned that prions will accumulate in the muscle. They're at much lower concentrations in the muscle, and the way I think of CWD as it progresses through the body of an animal is generally they're being exposed from their mouth or their nose through contact with infectious secretions from another animal. And so the front of their body gets exposed first. And so that's why we'll test those lymph nodes that are behind the jaw, that are draining the area of the mouth and the nose. Because that's where the prion's accumulating first in the tissue. And so that's the earliest tissue for detection and for reliable detection. It'll eventually move to the back of the body. And then over time, as the disease continues to progress and those prions self-propagate, it'll accumulate in the brain, creating plaques that lead to the neurologic signs, the neurological degradation. And so and that's why the delayed onset of clinical signs. But to get to the muscle question, so we know that the muscle can test positive, but we also know that it takes time for it to accumulate there. And without knowing, where your deer is in that stage, like there's a time where a deer can be infected that it's not going to test positive in the lymph node. It's a pretty short amount of time, fortunately for us from a surveillance perspective. But I personally have much lower confidence in, using muscle unless you knew that your deer was positive and then you would have already had the result to begin with. So that's kind of my perspective. But we've had people who, they disposed of the head and they wanted to test, and it is, you can definitely find prion there. I think there's just a lot more uncertainty about, how far along a deer has to be in its disease progression for it to test positive.
So a couple similar. Could you talk a little bit about EHD and how it's why we're seeing a lot of that right now? And
Yeah.
also, what do we encourage hunters to shoot sick-looking deer?
Okay. Um, so with hemorrhagic disease, so this could be a virtual public meeting in itself. So hemorrhagic disease, I'll kind of list out the differences maybe with chronic wasting disease. So it's a seasonal disease. It tends to affect deer during the late summer into early fall. And occasionally when we have warm-ups, we'll see some lingering mortality out until, the late fall. Which we've had some last year, and we had some of that this year, that lingering mortality. And so hemorrhagic disease can be one of two sister viruses, either blue tongue virus, which is generally considered a disease of domestic ruminants, namely sheep, but it can affect goats and cattle. And then epizootic hemorrhagic disease, which is generally considered a disease of white-tailed deer that are extremely susceptible in Iowa. But in other parts of the country, like in the Southeast, in Georgia, you can have outbreaks, ongoing, and you won't have the carcass count to show it. They'll be kind of asymptomatically affected or minimally affected. So in Iowa, it's still a pretty new disease. Our deer usually don't have immunity to it. Maybe if there's some natural selection that has to take place, that hasn't occurred yet. So most of the time, we think that our deer will just present as dead if they are exposed to these viruses. These are viruses, they're not prion diseases. So chronic wasting disease, prion disease, hemorrhagic disease, a virus that's actually transmitted by an insect, so a biting fly. And so kind of the whole disease ecology, how it moves through an animal, kind of what it does, it affects the blood vessels, the lining of the blood vessels, and creates, just a lot of leaky vessels. That's why you'll see foam and blood tinged foam at the nose sometimes if you have a recent mortality. And a lot of times these animals are seeking out water because they get very hot from all that damage that's happening to their vessels. So they get fevers and then they seek out water to cool down. And so a lot of times they'll succumb while they're in a pond or some other water body because they're cooling down from the extreme damage that they're facing from these viruses. So that's kind of the big kind of broad strokes of hemorrhagic disease. In comparison to CWD, like you can see hemorrhagic disease on a landscape. You will see dead deer. They get exposed and about 7 to 10 days later, they will become acutely ill and they will die within 36 hours. And so if any of you had experiences like that, where you saw a sick deer, a deer that was suddenly down dead in about a day and a half, that would fit hemorrhagic disease, especially if it was near water or definitely if it was in a pond. It's like pretty predictable. Whereas chronic wasting disease, it's insidious. We don't know that it's happening. We're not going to see a pile of dead deer. Whereas there have been properties that have been severely affected with hemorrhagic disease, but it's also a disease that we expect to hit an area hard and then it will go away. And maybe in 5 years, it will come back. And maybe with more severe weather patterns and things like that, we'll see more hemorrhagic disease activity, but then likely we'll also see deer exploiting, survival strategies. So, having some level of immunity, and so surviving into the next year where they don't get hit as hard, and that kind of thing. So, by comparison, a deer with CWD, it's 100% fatal. And that's across all the prion diseases, including those that affect humans. Once an animal starts on that path of having a prion disease, it will succumb to that disease, unfortunately. It generally doesn't go away from an area. So there's been very low success really eradicating the disease. We could point to kind of one case in New York where they had a detection. They intervened with a pretty heavy hand and were able to seemingly excise that infection. But in places where it's established, it's here to stay. So hopefully that gives a little bit more color and perspective.
Being a herding animal, especially in the winter, how does harvesting more animals and limiting the herd actually control the spread of CWD? They will still congregate the herd in herds and be in close contact with each other, whether there be 50 or 25.
Yeah. So the best way to limit the spread of CWD is getting those diseased deer off the landscape. And so how we do that is through harvest. Um, a diseased deer will have years to spread it to other deer. So natural behaviors like yarding behaviors, obviously we can't, overcome or change that kind of natural history of these animals, but we can change the unnatural ways that we have them congregate around salt blocks, these kind of high risk activities that are human mediated. So I I fully understand, and the behavior of deer is a challenge to managing this disease. But I think there's a lot of good we can do by changing human behaviors first.
We also would have less dispersal if we have less deer on the landscape. I mean, yeah, you get it in a family group, they're probably all going to get it in that family group. But the more likely, if you have lots of deer leaving and dispersing, obviously that's going to spread it quicker. True?
Definitely. So the strategy in other states where they use things like sharpshooting, they'll remove whole family groups for just that reason, because the risk of exposure to those other dependent or independent offspring will be high if we know that disease is in the area. But in other disease systems like tuberculosis, bovine tuberculosis, we know that limiting the natal dispersal. So if you remove a doe, her, buck fawn will stay in an area tighter than if she was still there. So it's all a balancing act.
Any anything you want to talk about as far as threats to livestock, either way, from CWD or
Yeah.
vice
So I did
versa?
see that in there. So there have been attempts to assess the species barrier, as it were, with cattle, which would seem like the obvious kind of concern with salt blocks being used commonly for cattle to supplement, trace minerals and that kind of thing, and then deer, overlapping in habitat. Um, and so the studies to date have relied on that method that I mentioned, the unnatural method of brain inoculation. So really forcing that species barrier. And they have found that cattle will develop clinical signs of disease after that brain exposure. And if you cycle it through, if you kind of take the brain of one of those sick cows and you cycle it back through, it'll affect more cattle and affect them quicker the next time they're exposed. So I kind of referenced how prions can change over times, and as they cycle through new individuals, what comes out can be a lot more effective at infecting that species in the future. And new species potentially. So it's a really complicated cycle. If that does, pique your interest, I'd say CWD ambassadors will really go over that. So please reach out on that front.
There's one question. Sampling time ranges from 6 days to 2 weeks. As a question, and I'm not sure if the question is how long can we take a sample? How long can a carcass be around before we can not get a viable sample, or why the disparity between results? Is it? So I'm not sure what exactly they're asking.
Yeah. So I can at least say if we can find the lymph node, we can test the deer. So we test some pretty gnarly roadkill in the summer. And as long as we can find that tissue, we know that the prions from the literature and other diseases and CWD, that they persist for years. So it should definitely persist one season in a roadkill animal, for instance. So if you have a head, we can pull that sample in most cases, unless it's a headshot and just the tissue is really damaged. So, and we just can't find it. So that's something to consider. In terms of the turnaround time, like I mentioned, with the hunter submission pathway, those times are more like a day to 2 days. But we do strive to have a maximum of 2 weeks from time of receipt. But I say that meant to emphasize that if you use, like, let's say the drop off freezer location somewhere, and that freezer had just been serviced that morning, it might be that there's a couple days lag where, before a staff person comes back there to check for heads. So there could be some additional days in there, even a week in there, just from lags in collecting the sample or sending the sample. But we're really trying to hit that 2 week period on the lab side.
Right. If the deer tested and found positive still appeared healthy, does this mean they were in the early stages? Also, can you tell from testing how long a deer has had CWD?
Yeah, that's a great question. So it is hard to interpret some of these things. If you were able to collect multiple tissues, so I mentioned that front to back process. So there's something that actually is used as a live test in captive cervids, using the rectal mucosa that has some of that good lymphoid tissue that we're targeting with the lymph node. You can actually kind of stage that disease progression, but without having multiple tissues in the same animal, it is hard. One thing, like, we use a screening test called an ELISA, an enzyme-linked immunosorbent assay, and we, kind of internally will say it's a hot reactor when it has a really high value, and a weak positive when it's a really low value. But that could be due to other factors like genetic traits of the deer or even prion strain factors. So there are different types of CWD. In Iowa, it seems like ours is kind of indistinguishable. So we wouldn't maybe expect it to be the prion strain side, but there could be some genetic factors at play, like affecting susceptibility or intensity of infection. But unlike scrapie, which is that prion disease of sheep, CWD, so far, we don't see any kind of genetic resistance to lean on. So the captive industry does really turn to, trying to breed it out of their population, which was very effective actually with sheep in the National Scrapie Eradication Plan. But in deer, it actually just delays the onset of clinical signs. So they become infectious for longer, which then means that the rest of your herd is probably getting exposed to.
You develop genetic resistance over time.
Well, I think we just hit on that. So to date, we don't know of that being a viable strategy. That would be one of those silver bullets, like, well, at least they can develop resistance. But so far, basically every animal that contracts a prion disease across species, across diseases, will die of that prion disease.
All right, I apologize if I'm asking questions that you just answered. I can't read and listen at the same time, so so I'm not doing a good job of listening to you. Are deer tested before they go into the Hush program?
So they could be, but they are not uniformly. That's not a requirement of the Hush program at this time. There are states around us that require it, or deer that are recovered from sharpshooting efforts and things like that, that those deer are tested before they are donated to food banks. But that is not the current practice in Iowa.
All right, of positive deer detected, what percent were bucks?
Of positive deer, so I would say this really, positive deer, bucks versus does. So we lean on that kind of who's at highest risk when we're getting those early detections. So like that first detection, or disease or CWD is new to an area. Once it's established in an area, really it's an equal opportunist. It can affect bucks or does because the sites that they're feeding on are likely contaminated. And so that environmental reservoir and chance for environmental spread or indirect transmission really goes up. And so in early in those surveillance areas, concentrating on bucks is helpful. But we've had actually several counties. Prior to last year, we had half of our counties, the first detection was actually a doe. And that could have been even a natally dispersing doe. So a doe that was leaving after her birth area in her first year. We just don't know, we don't have that resolution. The epidemiology of this disease is very hard. But I would say I don't think about bucks and does as much once we're talking about endemic areas like Corydon, Harper's Ferry, Elkader, those kinds of areas.
What is the difference between EHD and blue tongue?
Okay, so just another, just talking about those two, they're sister viruses. So they're very similar, just EHD primarily is deer are considered the primary reservoir. With blue tongue, sheep are considered the primary reservoir, but there's definitely crossover. So this year we've been seeing actually quite a bit of blue tongue. And we've had detections of blue tongue in the past. It's just usually it's the disease that we know affects deer, which is epizootic hemorrhagic disease. So it's an interesting trend we've seen this year, but we know that they can cross over. These diseases can affect bison. We know they can affect pronghorn, obviously outside of Iowa. So if they're in an area, the host range is pretty similar and overlapping between those two viral diseases.
Animals in Iowa, other than deer, cattle, or sheep, tested positive for prion diseases?
Other than deer, cattle, or sheep? Yes. So humans have several different prion diseases. So Creutzfeldt-Jakob disease, a lot of times in public meetings, there'll be someone who says, like their neighbor died of CWD, of chronic wasting disease. And what they're generally talking about is actually Creutzfeldt-Jakob disease, which is a primary prion disease of people. It has a spontaneous presentation. So, people, and usually it affects people in their later years, but it can spontaneously occur and then it self-propagates like we kind of described with prion diseases and causes that fatal infection. There's other ones, Kuru, that affected Polynesian people. Fatal familial insomnia is present in some that's a genetically inherited prion disease. And then there's, related to the mad cow disease outbreak in the 80s and 90s, there was spillover into other species, which included people. That was called variant Creutzfeldt-Jakob disease. It also spilled into cats, and there was a feline prion disease that was diagnosed, and likely a mink-related disease from feeding infected meat from cattle or contaminated meat from cattle. So it definitely can affect other animals. It's a hard disease to characterize. And so the resolution of data to know that you're seeing a prion disease, if it were to occur as like a one-off spontaneous presentation, it would be hard and likely missed.
Do you have any examples of states that have successfully managed CWD?
So that's an interesting question. That depends on how you define successful. So like I mentioned, New York is really the only case we have of a state having a positive, intervening, and then not having positives, any more positives to date. So that seems pretty successful, but I think that they intervened on maybe the very first case. And so all those other complicating factors like environmental reservoirs, and just number of infected individuals you had to figure out where they were was just so small in that scenario. There's states who have done a pretty good job, like Illinois has done a great job of keeping the overall prevalence low, but their local prevalence in counties since 2002 has increased, and they are moving away from sharpshooting as a sustainable activity. So, that's one thing that we really prioritize in Iowa. One, we partner with our hunters and public to get our samples. and actually to manage our deer. So we do have additional opportunities, harvest opportunities through an incentive hunt program in some of these endemic areas where we really deputize our hunters to achieve our disease management goals by harvesting those positive deer, or at least a pretty good fraction of them. Last year, we had our Dubuque County or Dubuque incentive zone for the first time, and we had a 24% prevalence rate. But that also means that all those deer that were positive are no longer able to transmit to the rest of that population. So really, harvest is our best way of managing at this point.
Rachel, if there was roughly 110,000 deer harvested in the state last year, do you have an estimate on what percentage probably had CWD out of that harvest?
That's hard to say. So I guess I can frame it like this. So we test about 5%, 5% of the total deer harvested. So I think it would be reasonable to extend out, maybe not a direct, a direct numerical, no multiplier to account for the other 105,000 that we didn't test. But definitely we know the CWD affects some of our really high density and high abundance areas, like in the Northeast. So we know that there's disease that we are not capturing in our surveillance data. So yeah, that's a good question. Everything's a proxy. We definitely know that there's more disease than what we are able to test for.
Return false positive?
What was the beginning of that?
What percentage of tests are false positive?
Yeah, so with our testing strategy, so we use a screening test called ELISA, like I mentioned. So basically what happens is a little piece of tissue from that lymph node gets tested. If it tests above a certain threshold value, it's considered a reactor, and then it gets retested. And the retesting involves taking a new piece of tissue from that lymph node and also retesting the original piece of tissue. And so we have 3 tests to base our screening off of initially. And then we confirm all of our samples with an additional level of testing through USDA called immunohistochemistry, which actually takes a slice of the lymph node and looks for the signatures of misfolded prion protein. And so really we're getting like 4 tests on every one of our positive samples. When we do have samples that come back as really low reactors on that screening test, oftentimes we'll also send in if it's right at that threshold where it's flagging and then on retest it's going below that threshold and being considered not detected, oftentimes we'll pursue the other paired lymph nodes. So there's 2 lymph nodes in the deer. We consider the one that we submit for testing the A, and then we bank another one that we often use for research or other applications. We call that the B. But when we need to use that to kind of confirm suspicions or things that aren't quite clear, we'll use that as well. So our false positive rate is really, really low. And nearly all of our positives have been confirmed on that confirmatory testing, which is definitely good news.
Is CWD a density dependent disease?
So there's aspects that are density dependent. So if there's lots of deer in an area and you have an infected deer in that population, it can definitely contribute to the spread of disease, but it also has some frequency dependent aspects. I really don't lean on those kind of descriptors often because, there's places, Greene County, Grundy County, where we've had detections. They're still just those single detections. But those were not high density areas. And so they would kind of laugh in the face of the density dependent process. But it's a good question. And it definitely has a role in the disease ecology of CWD.
blockers on testing to prevent the spread?
Lockers.
Blockers, yeah.
Um, so, in conversation with IDALS and their Meat and Poultry Inspection Bureau, they really consider the standard cleaning practices of equipment to be sufficient for CWD. And a lot of what that involves is keeping equipment wet so things aren't adhering. So, like if there was an infected deer to come through, its tissues aren't adhering down to the metal equipment. And we know that prions have an affinity for metal and for binding to those kinds of surfaces. So really that's like what the focus is just keeping things clean and kind of and wet as much as possible in between processing animals. So on that front, you know, the amount of prion that would be in the meat of a positive animal is, I mean, likely it's just a drop in the bucket, compared to like a deer having direct contact with another deer or something like that. So lockers are definitely one of our partners, but they're not key to limiting this disease.
Suspecting that the first CWD was detected in Colorado and Wyoming, how do you explain the sporadic spread across the Midwest region, and what are states doing to slow this down?
Yeah, so it's hypothesized at least that the way CWD made its way into the Midwest likely wasn't through the movement, a great movement of a individual animal, but actually through humans hunting deer out west and bringing them back. And of course, not knowing what chronic wasting disease was. So it was first characterized in 1967, but the disease-causing agent, being a prion, wasn't known until the 80s. So all this was playing out for decades before, anyone knew that there was anything wrong. So likely it was that harvest of positive animals from out west that brought it into likely, at least Wisconsin and Illinois. And then it spread from there locally. Like I mentioned, carcasses can spread disease. They're infectious, and deer are known to, they'll inspect carcasses, they'll chew on bones for calcium, that kind of thing. So we know that that's a viable route of exposure to these animals. And how it's moved since then, that's why the epidemiology is so complicated. So we know deer move. We definitely know, we have elk that come through Iowa all the time. So we know that there are cervids moving on our landscape, and they can move quite large distances. Some of the new collaring data out of our neighboring states shows, huge movements of even females, which were kind of thought to not be the big movers and shakers in disease spread. But even yearling does will move, 70 kilometers, and things like that. So that's one aspect. And then the human mediated aspect, of course. So just more people kind of harvesting deer in some of these positive locations, maybe unknowingly, maybe knowingly, but not realizing that what they were doing could cause harm in, their home location. And based on that map, we have a lot of movement of deer across the state, and there's comparable maps that show movement across the country. So people unintentionally are helping the spread of this disease for sure.
All right. Captive deer breeders appear to be making great strides with CWD and deer survival by using humic acid as a feed supplement. Have you heard about this? Is that a viable option?
Yeah, so there is a lot of research. I mean, one thing I could say is that the amount of attention that CWD is getting in recent years it's grown astronomically, and from a lot of really interesting groups. There's soil scientists figuring out where clay substrates exist, and if that explains where we're seeing outbreaks of disease. Humic acid for sure is being discussed with some of the chemists and that kind of thing. They're all in the conversation. I would say anything that's turning to a feed supplement, much like, even a vaccine, there's conversations, and there are research groups in Canada actually working on a vaccine platform that could really possibly change the game. But deploying it to a wild population is always going to be that challenge, like just a challenge that we can't, discount, because deer are wide ranging, and getting anything out, whether it was a food supplement or a vaccine, would be incredibly difficult, on a free ranging population of wild animals. Whereas in a captive setting, you can explore some of those options to see, um, you know, decreasing the burden of disease in an area or the amount of infectious prion protein within a confined space.
The question is, would CWD be worse in dry years? Would the prions be more easily kicked up in the dust and then ingested? Anything in particular you would say about weather conditions or drought or anything like that would have any effect on CWD?
Yeah, that's an interesting question. One thing that I actually think is maybe helpful in Iowa with how much tilling there is and how much turnover of soil that might bury prions. So that's not even weather related. That's just, being in an agricultural state. That might actually help make prion less accessible on the surface. But by the same route, the duff in a forest, as leaves degrade, they're covering up prions. So unless it's being continuously deposited, that can really be helpful, for controlling, access to those prions. Um, but anything that's really changing the patterns of deer. So if, I could kind of play out a scenario where, we're in a drought year, deer are seeking out water sources, maybe like leaning more on bird baths in urban settings or something like that, where, contamination of the water source itself, much like, a supplemental feeding station, could actually, contribute to spread. So, yeah, I'd have to think about that more. The dust is an interesting question.
We've hit most of them, and we are 15 minutes past 8, so maybe we should let it go. And Rachel, before you log off, maybe you want to look at through these questions and answer any you can in writing. And we
Yeah,
can have that included with the what's posted.
absolutely. And like I said, for anyone who's still on, please email me if you have questions. I talk about CWD, or at least think about CWD every day. So I'm happy to talk about it further with you, on a one-on-one basis. So thank you again for joining us.